All right, let's get into it. We're going to explore one of the most critical parts of clinical research, figuring out who exactly we should study. And what's interesting here is that it's not really about a strict set of rules. It's much more about a way of thinking, a kind of art, really. It's a constant balancing act between the perfect scientific ideal and what's actually possible in the real world. And that brings us to the fundamental question that drives everything, doesn't it? How on earth can a study of, say, a few hundred people tell us something meaningful about millions? Well, the answer isn't magic. It's actually a really careful, deliberate process of starting with a huge, broad idea and then slowly, thoughtfully, narrowing it down to a manageable reality. You see, every study starts with this big picture group in mind. We call this the target population. It's the complete group of people that a researcher hopes their findings will eventually apply to. It's a concept, really, defined by things like clinical traits or demographics, something like all teenagers with asthma. But of course, you can't study all teenagers with asthma everywhere all at once. It's just not possible. So that big idea has to meet reality. It gets refined by practical things, like geography and time. This gets us to what we call the accessible population, that subset of the target group that's actually within a researcher's reach. So maybe that's teenagers with asthma in our city this year. From that more manageable, accessible group, the researcher then has to make an even more specific plan. The intended sample is the group they actually aim to recruit, and that's often shaped by who they can practically contact. So now we're thinking something like teenagers with asthma who are treated at the 3 local clinics we can actually partner with. And this brings us to the final step in this whole funnel, the actual sample. This is who really ends up in the study. It's only those individuals who are contacted, who meet every single bit of the criteria, and who, at the end of the day, agree to participate. It's the end result of a long chain of choices, and honestly, just circumstances. So, you can see how this whole funnel creates a pretty big challenge, right? How much does that final group of people, the actual sample, truly represent that huge target population we started with? Bridging that gap, it's not a simple calculation. It's an art that requires some really careful judgment. A classic example of navigating this is the Framingham Heart Study. The sample they used was definitely not representative of the entire U.S. population at the time. And yet, the associations they discovered, like the link between high blood pressure and heart disease, have turned out to be incredibly generalizable. The big idea here is that even a sample that isn't perfectly representative can still lead to valid conclusions. But, and this is a big but, judging how far you can stretch those conclusions takes some deep, thoughtful consideration. And just like the researchers in Framingham had to define their group, every single study has to precisely craft who belongs and who doesn't. This is all done using what we call selection criteria, a tool that lets researchers shape their study sample with almost surgical precision. But this reveals a huge tension that researchers are always dealing with. It's this constant balancing act. You need your criteria to be narrow enough so you can isolate the specific effect you're studying, but at the same time, it has to be broad enough for the results to actually matter to a wider group of people. And on top of that, it has to be practical. Can you even find these people? While also being fair, making sure you include a diverse range of participants. Let's try to make this a little more concrete. Imagine a team wants to test out a brand new fitness app, and it's designed specifically for people over 65. First, they have to decide exactly who to invite into their study. And here's where it gets tricky. This is their dilemma. If they only include seniors who are already tech-savvy, well, the data they collect is going to be clean, it'll be easy to analyze. But would those results really apply to the much broader population of seniors, a lot of whom might struggle with new tech? It's a classic trade-off between getting really high-quality data and making sure your findings have real-world relevance. That whole fitness app question is a perfect example of another tension that just runs through this entire process. And that's the constant negotiation between the most scientifically rigorous, perfect methods and what a research team can actually pull off with their limited time, money, and resources. The chapter really brings up 2 main ways of thinking about sampling. The gold standard, the ideal, is what's called probability sampling. That's where every single person in your accessible population has a known random chance of being selected. It's beautiful because it allows for powerful statistics, but it often requires a complete list of that entire population, which can be impossible or just wildly expensive to get. And that's why so many studies end up using non-probability sampling, like, say, just enrolling patients as they happen to walk into a clinic. It's way more practical, but it means that generalizing the results relies so much more heavily on the researcher's own careful judgment.
So we've got all these theories about sampling, but then there's the very human and often very messy reality of actually convincing people to sign up for your study.
This quote is it's incredibly powerful because it captures such a sobering truth. Failing to recruit enough participants is one of the most common and, frankly, emotionally draining problems in research. It's a really harsh reminder that a perfectly designed plan means absolutely nothing if you don't have actual people willing to take part. So, why are we spending all this time on this? Why does it all matter so much? Well, it's because every single one of these early decisions about who to study and how you're going to find them has consequences that just echo through the entire research process. Right, so the crucial point is that if these judgments are made poorly, the entire study can be compromised. For instance, if only the healthiest people volunteer for your study, that's called volunteer bias, your results might get skewed and look way too positive. Or if certain groups just don't respond, a problem we call non-response, then your findings might not apply to them at all. At the end of the day, flawed sampling can lead to seriously flawed conclusions that can misguide both science and medicine down the road.
One message that comes through again and again is that choosing your study participants is not like following a recipe. It's a really complex process of negotiation. It's a whole series of thoughtful trade-offs, trying to find that sweet spot between scientific purity and what's actually possible in the real world.
In essence, these early choices about the study sample, they lay the foundation for absolutely everything that comes after. They actually determine the limits of the study itself, and they define the scope of the conclusions that can be made at the very end. Getting these choices right is what truly prepares a researcher for the entire journey ahead. In the end, what is the story a study will be able to tell? Well, that story is written, in large part, by who is invited to be in its pages.